understand the basic characteristic of the bacteria, and the last one, know the important structure of the bacteria. So for the way that we use for classify the bacteria, we can determine it like by varieties of the criteria. No? So we can determine it by shape, their arrangement, their staining, their oxygen requirement, temperature requirements, mode of nutrition, or the thing that they use to survive. Serological system, so we see from the antigen, the protein outside surface of the cell wall. And also sometimes we can classify by spatial structures of the bacteria or spatial features. And the last one is the genotyping system. So we use the information of the DNA sequence to classify or to grouping the bacteria. No? So for using the shape and arrangement to classify. So for the first one, most of the supragingival plant microbe we compose of the complex bacteria, which is the shown in spherical shape. So for the bacillus or the bacilli bacteria, so it's shown in short, in rod shape. So sometimes it's shown in short rod or long rod. So and the other one is spirogyrum, or group of the spirochete bacteria. So it's shown in spiral shape. So this shape of the group. So if we mention about some examples in our oral cavity, most of the bacteria that relate to severe periodontitis will have like spirochete shape. And also we have some different type of other shapes, such as fusiform, that we also found in our oral cavities too. Coccobacilli is a very short rod. Now, so it's like showing like this type of shape, vibioid and helical. These two types we quite not found in our oral cavities. Most of the time we found just cocci, bacilli, spirochete, fusiform, and coccobacilli. Why the bacteria need to have different shape, no? So actually it's related to the survival skill of those bacteria. So it's like sometimes if we change the condition of or the environment of the bacteria, they also change the shape too. So sometimes it's the chemical or the chemical in the environment will trigger the changing or shape of the bacterial shape. Nutrition variable is also involved. If it less food, so it might be like shorter, or sometimes it might be longer. It's up to the conditions. So the selective force that involved, the first one is the predation. Why the predation is involved? Because in the environment, so in the predation normally we found in the not in our human body, normally we found in lake, in ocean, and so on. So we have predator and prey. So once the predator need to eat that prey, so they have to have a proper size, right? So if you very big and you eat a very small, you put the energy to eat something very small, so it's not gain energy, equal energy back. So sometimes in the close environment, the proper predator and prey, the predator will try to hunt the prey that appropriate size to when you compare to the energy that you put in. So like in this case, so in the so then once in here, the first picture is like in the normal environment before we put the predator in. And after we put the predator in, we found that some side of the microorganism is lost, because in this side is like the side that proper for the predator to put energy to eat. So if we leave the condition like this, normally the size that proper for the predator to hunt will be reduced, right? So it will be the selective force. So it will be like the prey that left will be like very small or too big to eat. So here is one selective force type that we might find in the environment. The second one is the nutrition in this selective force, we also found in the human too. It's like sometimes if the bacteria they stay in the area that lack of food, sometimes they increase the size to increase the surface area that the cell can that the molecule can absorb in เหมือนกับว่าบางครั้งในกรณีที่ธรรมชาติมันแบบโหดร้ายมากๆมีอาหารน้อยถ้าเราตัวเล็กๆเรามีพื้นที่ผิวด้านนอกน้อยเรามีโอกาสที่จะแอบสอบได้น้อยคราวนี้เชื้อบางชนิดมันก็จะยืดตัวให้มันยาวขึ้นเพื่อให้มันมีพื้นที่ผิวในการทั้งเกาะด้วยแล้วก็ให้มันแอบสอบได้มากขึ้นอย่างเงี้ยก็ได้นะคะสำหรับ for the nutrition เนาะ and for the dehydration is sometimes it's very dry so they reduce the size to reduce the surface area to like let them stop losing the moist นะคะ so and also the attachment if like you stay in the spherical shape is quite very easy once when you stay in the environment that like have a water flow to flow you might really easy to eliminate from the environment so they increase the size to increase the area to attach onto the surface นะคะ so here is like four major selective force that can make the bacteria change their own shape So in this example is the E. coli that cause uropathogenic. That is the uropathogenic E. coli นะคะ that cause disease in our kidneys นะคะ So in this case, normally the bacteria, the E. coli will be a short rod shape, no? A rod shape, not short rod shape. Once they invade into the host cell and they want to escape from the white blood cell to eat them, so they increase their size into filament form to make sure that the white blood cell cannot eat them and then they can survive in the environment like in our host bodies. So here is some protection mechanism that bacteria use for fight with our immune systems. And here is the arrangement for the coccyx or coccyx shape of the bacteria. So we two main arrangement that you need to remember. Which is the chain and cluster shape. So in chain we call it streptococci. Why we need to remember? Because this will be like present in your examination for sure. Even if it's not in my quiz or my test, this will be present in NL test. So we would like you guys to distinguish between streptococci and staphylococci. So because the streptococci is the major group of the microbe in our oral cavities, and staphylococci is also cause disease in nasopharyngeal tract. So for the streptococci group, so the way that the plane of division is like in just only the middle one. So after they do the binary division, so they just like increase the number in the linear forms because they have just only one plane. But for the staphylococci or the cluster one, they like have multiple plane for cell division. So it's like once they finish divide, they will see in the cluster look like the graph ones cluster. And for the bacilli, so we have just chain of bacilli and single ones. So we can call this, if it just do binary fission and the two cells still stick it together, we call it diprobacilli. But if it stick between each other more than two, we call it streptobacilli. In one cell and very short, we call it coccobacilli. So now we go into the ground staining of the bacteria. So we use differential staining in this case for determine type of the bacteria. So the structure that involved in this ground staining is the cell wall of the bacteria. So gram-positive and gram-negative bacteria have different composition of their cell wall that lead to different way that they stain by the colors. And also the acid-fast bacteria, their cell wall also different from the normal type of the bacteria, which we will talk about after this. And the other way that we grouping classify the bacteria is according to their oxygen requirements. So the anaerobic bacteria is the bacteria that need oxygen for their survival. Aerobic bacteria. Facultative anaerobic bacteria is the bacteria that that require oxygen for their survival but can grow without oxygen. So a lot of oral microbes, they grouping as the facultative and aero. So they can survive in oxygen, but actually without oxygen they still can grow. Microphyllized group, so this group require oxygen but at a low concentrations. And the last one, which is the obligate and aero, so they cannot grow in the place that present of the oxygen. And so most of our subgingival microbe will be classified in the group of obligate and aero. So why the classification by oxygen requirement is quite related to our fields, because actually in our oral cavity they provide a different type of niche or different type of ecology system. As you know that if we call it supragingival plaque, that means the group of microbes that forming above the gingival line, right? So in that area, the oxygen can access, right? So the group of the bacteria, which is aerobic bacteria or facultative aerobic bacteria, they can grow on your teeth, on your tongue, on your buccal mucosa. But if you talk about the place under your teeth, under your gingival, which we can call subgingival plaque, in those places is the place that have less oxygen. Sometimes if you go into quite a deep of your gingival, so it's in that area is like no oxygen anymore. So the group of the microbe already changed because different type of microbe need different volume of the oxygen, right? So in this area of our oral cavity can provide varieties of concentration of oxygen. So this type of classification quite important for us to know too. And this way to classify is quite not related to us at that much, but is quite related to environmental microbiology more. Because most of the bacteria that we will learn in this subject will be like normal flora or pathogen. So normally it's grown at 37 degrees Celsius, which is mesophyte. And the last is nearly the last one, the way that we classify. So we classify from biochemical activity of the bacteria. So we observe the metabolism of the bacteria that they might produce some metabolite or secondary metabolites or some special enzymes. After they use those precursors and they can produce metabolite, so we can determine the metabolite on the agar that we grow the bacteria. Then we can use it for classify the group of the bacteria. And for the other one is the serological, so we determine the protein on the cell surface of the microbe, and then we use the specific antibody to grouping them. So if they're specific to those antibodies, so we can identify the type of those bacteria too. And as I said in the identification part, so we can determine the sequence of the DNA to grouping the bacteria too. In this case, we use the ribosomal DNA for determining the type of the bacteria. And the spatial structure, like spore-forming bacteria, non-spore-forming bacteria, or sometimes we count the number of the flagella on the bacteria cell to grouping them too นะคะ So next the bacterial structures so we have external we will talk about the external structures นะคะ So the external structure compose so we will group into T type the external structure internal structure and the part of the cell envelope So the external structure will compose of flagellum, pili, fibrils นะคะ So these three appendage is involved in motility of the bacteria no? And the last one and the next one is the glycocalyx can be grouped into capsule and slime layer no? These glycocalyx are involved in the cell protection of the bacteria นะคะ For the appendix part no? So the appendix part, as I said, that is involved in the motility of the bacteria and also the attachment. The structure that involved in motility will compose of flagella and pili. For the attachment or forming the channel to communicate with other bacteria, which will be fibril and pili. The first one I will talk about is the flagellum นะคะ So the flagellum is the long filament structure that protrude out from the bacterial cells no? So it's longer than pili นะคะ So the purpose of flagella is involved mainly on motility of the bacteria no? And chemotaxis. So what is the chemotaxis? The chemotaxis is the reaction that bacteria respond to the environment stimuli or chemical stimuli นะคะ So is it food? The bacteria will use their flagellum to swim to the food. But in case if it's toxic, they will just swim out on the area that are toxic areas นะคะ We will call it chemotaxis. No? And here is the way that we grouping the bacteria according to the number of the flagellums นะคะ They have each one have their names, but no need to remember. No? And for the flagellum, the flagellum have three major parts, which is the first one, filament part, hook part, and basal bodies. No? The basal body is the part that they attach to the cell membrane of the bacteria. No? So they help to move the flagellums. And the hook part is the part that So is the part that connect between basal part and the filament part. The filament part is the part that they use for controlling the movement. No? Actually, it's the video of the swimming of the bacteria by flagellum. Okay, it's the word. So here you see it look like prop school one when the hook part is moved, so the flagellum will move too. The second one is the pilars นะคะ Pilars is like shorter than flagellum no? And they have variety of function from the pilars no? So they can use from host adhesion no? The host adhesion is quite very important because before bacteria can infect into the host cell, they have to adhere first. So the pillar is quite very important in this reaction. And after they can adhere on the surface, they start to do the colonizations. And it's involved in biofilm formation and also DNA uptake. That was the function of pili too. No? So the biofilm formation is quite important for the dentist because all the plaque are the biofilms. So the pillar structure is quite important. No? The way that bacteria move by the pillars, the way that they move is quite a bit different way from the flagellum move. No? We call it twitching motility. No? มันจะดูดึบๆหน่อยและจะไม่ค่อยเหมือนflagellum motile นะคะ Okay, the pillars is composed of pillin proteins นะคะ and they have some different structure of the pillars between gram-positive and gram-negative bacteria. No? The way that the covalent of the pillin protein is different. So the type of the pillars, no? For the type 1 and type 4, they use for adhere onto the surface, but they also contain the pillars that cause set pillars. Now, these pillars is quite important because they are used for transfer the genetic material from one bacteria to the other bacteria. So here is the way we call it horizontal gene transfer. It's quite different from the binary fictions now. So sometimes the set pillars they can transfer the genetic material of different species of the bacteria too. Sorry, we have a picture too. So here is the set pillars. Once the donor wants to transfer the DNA or genetic material to the other one, they just form the pillars and then we can transfer it. The last one is fimbrium. Now, fimbrium was composed of the protein that we call fibrin. No? So it's the smallest one and the thinnest one. Once when we compare to the flagellum and pillars. The fact that function is only used for adhere to the whole cells, no? And this lower table is used for comparing the difference between flagellum, pili, and fimbriums. The next one of the external structure is the glycocalyx one. No? So the glycocalyx one will be grouping into like slime layers, no? Which is called the glycoprotein that loosely associated with the cell wall. So for the function of the slime layer is they use for attach onto the environmental surface. No? It's because it's slimy, it looks like a glue, no? So it's help to attach. But in case of the capsule is the glycoprotein that firmly attached to the cell wall, and the capsule. And the capsule is the repeat polysaccharide and protein that accumulate on the surface of the bacteria. So the capsule is quite very important because it helps to protect the bacteria from the harsh environments and also for the pathogen bacteria. So it helps to resist of the phagocytosis. So it helps to escape the immune of host for the capsule one. For the cell envelope, composed of cell wall and cell membranes. Cell wall is the structure that determines the size, the shape of the bacteria. So it helps to protect bacteria from the osmotic pressure from the environment. So it's a rigid structure that protect the organelle inside the bacteria. And the main important thing is the peptidoglycan is the chemical that is the structure of the cell wall. Peptidoglycan is the glycan chain link with the short peptides. And it's quite important because we use this for bacterial classification. Gram-positive, gram-negative bacteria, we determine by the composition of the peptidoglycans. And also, once you learn in pharmacology subjects, you will know that here the cell wall or the peptidoglycan is the main target of many types of antibiotics. ก็คือตัว peptidoglycan เนี่ยสำคัญมากเลยเพราะว่าตอนน้องๆเรียนวิชาเภสัชกันก็จะรู้ว่ามันจะมียาหลายตัวมากที่มันจะ target ที่ peptidoglycan แล้วมันให้ประโยชน์ยังไงเพราะอย่างที่เราบอกว่า peptidoglycan เนี่ยเป็นตัวสำคัญมากที่จะทำให้เซลล์มันเนี่ยเป็นเซลล์อยู่ไม่งั้นมันจะโดน osmotic pressure ทำให้เซลล์แตกเพราะฉะนั้นถ้าเราทำลาย peptidoglycan ได้เซลล์แตกเชื้อเป็นไงคะเชื้อตายใช่เพราะฉะนั้นยาหลายตัวจะทำเก็บตัวนี้ก็เลยจะค่อนข้างสำคัญนะคะสำหรับ peptidoglycan So now we move to how the peptidoglycan can classify bacteria into gram-positive and gram-negative bacteria. No? For the gram-positive bacteria, they have a very thick peptidoglycans, like you see in this picture. The purple one is the size of the peptidoglycan, quite very thick. And after the peptidoglycan will be the cell membrane. Contrast to the negative gram-negative bacteria. The peptidoglycan is quite thin. The pink color, the dark pink, chocolate pink here is the peptidoglycan of the gram-negative bacteria. It's quite thin And the other property of gram-negative bacteria is they have two membrane layers. The first one is outer membrane layer, and the second one is the inside membrane layer. So that means for the gram-negative bacteria, the peptidoglycan is like a ham or sandwich. So it stays between outer membrane and inner membrane. So in this property that make this peptidoglycan stay in different way. So here is the synthesis of peptidoglycans. Actually, for my lecture, it's quite not very important, but I think it's important for you guys during learning for the pharmacology part, because as I said that the peptidoglycan is one of the main target of the antibiotic that we use nowadays. So I just go roughly on this thing. So the first part of the peptidoglycan production will start inside the cytoplasm of the cell. After that, the precursor of the peptidoglycan will free transfer out into the area outside the cell membranes. And then they link to each other to cause the network of the peptidoglycans. This picture is the way of the gram-negative bacteria to construct the peptidoglycan layer, because you can see that here they have inner membrane and outer membrane. But in case of gram-positive, they will lack of the outer membranes. And the important enzyme that be the target of the antibiotic is transpeptidase. The transpeptidase, their function is they link the peptide together. Peptide that attach onto the glycan together. So normally the drug will stop the function of transpeptidase, which will create, which make the network cannot connect to each other, so the peptidoglycan cannot produce the big one. So here is the video of the cell wall synthesis. If you're interested, you can go to see by yourself. If not, that's fine. So here is the close look of cell wall, gram-positive bacteria cell wall. So like I said, we have the cell membrane, we have a very big peptidoglycans, and we not just only the peptidoglycan that we also have teichoic acid and lipoteichoic acid. These two types of the acid will help the cell wall more rigid. For the gram-negative bacteria, the structure is more complex. So they compose of two membrane layers, outer membrane, inner membranes, and the peptidoglycan is between the outer and inner membrane, which is quite thin. And they also have the periplasmic space, is the space that some synthesis occurs. The space between outer membrane and inner membrane. And on the outer membrane, they have the protein that we call porin. So this protein will create the pore of the outer membrane, so the some type of chemical can pass through that pore and then go into the bacterial cells, inside the bacterial cells. Lipoprotein is another type of the protein that enhances the rigidity of the gram-negative bacterial cell wall. But the other part that's quite important for gram-negative bacteria is lipopolysaccharide. So it also helps to make the cell wall more rigid, but it's involved in host immune response. This lipopolysaccharide or LPS is one of the major virulent factors of the gram-negative bacteria. So the LPS, you will see a lot after this during you learn about the pathogen bacteria, the gram-negative pathogen bacteria, because as I said, it's one of the major virulent factors. So LPS composed of three major parts. The first part is oligosaccharide chain, which contain all antigen, and this region is quite very variable. So different type of bacteria will have different type of polysaccharide chain. The second part is the core polysaccharide, so it's quite very conserved for the LPS. So we found this core structure, the same one with different type of gram-negative bacteria. The last one is the lipid A. So the lipid A is the major part that play role as the violin factors. So lipid A is the part that they embed into the outer membrane of the bacteria, and we call this as an endotoxin, because once this released into the host cell, so that it will be activate the immune response of the host cell. So this called endotoxin, the part of LPS, lipid A part. From the gram staining method, because as I say, the difference between gram-positive and gram-negative bacteria is the peptidoglycan, the thickness of the peptidoglycans, and structure of the outer membranes. If the bacteria grouping in gram-positive bacteria and gram-negative bacteria was aimed by gram staining, it will show different way in each step. So the first step, we put the crystal violet, which is the primary dye, onto the cell. After we fix the cell didn't contain any color, right? After we put the crystal violet, the cell both of the gram-positive and gram-negative bacteria will show in purple. After that, we put the chemical that we call iodine or gram iodine. This gram iodine will, in case of gram-positive bacteria, it will help the crystal violet to bind together with the iodine and form a large structures. Those large precipitation will trap inside the peptidoglycan network. And in this case, because the gram-negative bacteria, they have the outer membrane layer, right, and they have a quite thin peptidoglycans. So the structure for the precipitation structure between crystal violet and gram iodine have less space to trap inside, right? ก็คือใน gram negative bacteria เนี่ยมันมี peptidoglycan บางๆไอตัว complex ระหว่าง gram iodine กับ crystal violet มันมีที่ให้ติดน้อยกว่าแต่ในขณะที่ gram บวกจะมีที่ให้ติดเยอะกว่านะคะ So in this step we still see the purple colors นะคะ of both of gram positive and gram negative bacteria. But the critical step is the decolorization with the alcohol. So the alcohol will remove the excess color out. But in this case, because the big peptidoglycan of the gram-positive bacteria have a lot of network and the precipitation already trapped inside the network. So the color still remain inside. It didn't wash out by the alcohol. But in case of gram-negative bacteria, they have the outer layer, and so they have the very thin peptidoglycan, which is less space for the color to trap inside. So once we wash out with the alcohol, so the crystal violet will remove out too. เพราะว่ามันมีทั้ง outer membrane มีทั้ง peptidoglycan ที่มันบางๆพอตอนถูกล้างออกไปสีที่ถูกแทรปอยู่น้อยอยู่แล้วมันถูกช้าออกไปหมดตัวเซลล์ตัวนี้ gram negative ก็จะเป็นสีใสๆละนะคะ So after that we put the counter strain in this case, which is safranin. The safranin will show in like pink color or red color, and also the gram-negative bacteria that didn't have any color at this after decolorization, they will stain with the red color. So now we can distinguish between gram-negative and gram-positive bacteria. For the acid-fast bacteria, which is the major way to identify Mycobacterium tuberculosis. In this group of Mycobacterium, they have a bit different structure. So on their cell wall, they compose a lot of many types of lipid. So the major part is microlic acid lipid นะคะ So this type of lipid will be present on the cell wall of microbacteria. No? So here is peptidoglycan is a bit quite thin นะคะ compared to gram-positive bacteria, but the lipid part, which is microlic acid, is quite very thick. This will protect once when we try to stain the acid-fast bacteria. So we have to use a different way to stain it นะคะ So in these acid-fast bacteria, in acid-fast stains or cell stain นะคะ So we use the carbon fuchsin as a primary dye. So the carbon fuchsin will be present in pink colors นะคะ And then after that we need to heat the slide after we apply the colors, because as I said, in case of microbacteria, they have a microlic acid cell wall. The lipid part is quite very thick. Once we heat it, it will melt like the butters need heat, right? So it melt a bit, so the color can penetrate into the lipid layer, those lipid layers นะคะ And after we heat and let the dye go into the layer, so we can still see the color of the carbon fuchsin as pink, but the step of decolorization is a bit different. So for the gram stain, we use just normal alcohol to wash, right? But in case we use acid alcohol to decolorize, because the acid will remove out the excess colors that adhere on the surface of the cell wall. So in this step, once we use the acid to remove out all the excess color, so in this step, the acid-fast bacteria will still show in red because they have a very thick microlic acid layer. But for the non-acid-fast, the color will remove out. So we put the counter stain by using methylene blue to see. So for the non-acid-fast, the methylene blue will attach on the cell wall of those bacteria, then we can observe it by purple or dark blue colors นะคะ So here we use for identify the sputum of the people who infected by Mycobacterium tuberculosis นะคะ And here is the comparison table. The next one is the cell membrane. I'm pretty sure that you learn about the cell membrane a lot in biochemistry subjects. So let's just go briefly on it. So the cell membrane is the lipid bilayer. So they have two layers of lipid. The hydrophobic part will be inside, hydrophobic part will be inside of the tail, and hydrophilic part will be the outside of the cells นะคะ The cell membrane is the selective permeable barrier, so they can select something to pass through inside the cells. And in this area is the place that energy or ATP generates for the bacteria. For the internal structure, they are composed of cytoplasm, nuclei, ribosome, and endospore. The cytoplasm is like the pale blue here. Is the gel-like matrix. They are composed of water, enzyme, nutrients, gas of the bacteria, and here is also the place that cell metabolism occurs. In this cytoplasm, there will be the place that contains chromosome, plasmid, and other organelles of the bacteria. As bacteria are prokaryote, so they have only nucleoid regions, they lack of nucleus, as we know that is non-membrane organelles. And the chromosome of the bacteria are circular double-strand DNA. And the other part of the genetic material that bacteria have, we call plasmid. Plasmid is the small circular DNA of the bacteria. Normally, they carry not essential gene for bacterial survival. So the chromosome will carry essential genes for bacterial survival. But in case of plasmid, so the they carry the gene that like increase the properties, such as antibiotic resistant genes or the gene that produce some type of special metabolites will carry on the plasmid นะคะ The next one is the ribosomes. So the ribosome is the structure that involved in protein production นะคะ For the bacterial or prokaryote one, so they also have two subunits of the ribosome like eukaryote, but the sedimentation value is different. No? In case of the bacteria, they have 20. Once when we combine together, we have 20 sedimentations values, นะคะ and the two units that combine to be the assembled ribosome, they compose of 50 and 30 sedimentation units, no? To be a ribosome for produce the protein for the bacteria นะคะ For the bacterial reproduction, normal way is the binary fictions นะคะ So for the binary fiction, the bacteria just increase the cell elongate cell size and then they duplicate their genetic materials นะคะ and they divide into two นะคะ But in case of the harsh environments, sometimes the bacteria they produce the spore to resist the harsh environments นะคะ The way that they produce the spore in endospore is quite a bit different. So increase their size, increase their genetics material size, and then they divide into two. And after that, the mother cell. So we call the first cell as the mother cells, and then we call the spore cells นะคะ So after they elongate and duplicate their genetic materials, the mother cell will engulf the daughter cell or the spore cell to increase the cell membrane layer, to increase the thickness of the cell membrane layer. After that, the DNA of the mother cell will degrade, นะคะ and then they will degrade the mother cell out and release the spore to the environment นะคะ So here is the more detail of the spore formation or the endospore formation, no? But in brief, it's just like I was saying in the previous slide นะคะ And here's the clip of the spore formation. And actually, not just only the binary fiction or spore formation, some bacteria they can do reproduction in different ways. No? They might do buddings, and also they can like produce. They can do budding, or they can like increase the size, increase the number of cells inside the mother cells, and also they can do the fission, so like fragment the mother cell into the small to produce the daughter cells. So here is the other different way of bacterial reproduction. Not just, I would like to introduce you that bacteria didn't have just only binary fiction, but they also have another way too. The biofilm, which is the important for us, no? The biofilm is the community of the bacteria that live together. They adhere onto the biological or non-biological surface, นะคะ like this picture. So the dark orange one is the thickness of the biofilm that form in the tunnels. No? This is also the same way that the biofilm from plaque on our teeth is the same things, นะคะ So once the bacteria forming the biofilm, most of the time they produce the glue-like polymer to adhere each other together. So the biofilm will help the bacteria to resist from the diverse environments. Even in case of pathogen, they help to resist to the drug or host immune system. And here is the pictures of how the bacteria or the biofilm spread from one area to the others. So as we know that it's actually in our oral cavity is the proper place for the bacteria to grow because they have food, the temperature is 37, it's quite very moist, so it's proper for forming, and the bacteria they have sugar because you intake the food too, right? So they forming the biofilm, but actually because we move our mouth all the time, right? So it's quite very easy for our plant to like หลุดออกมา from the biofilms, no? So that means they can circulate into other organ of the human. So after this one, when you learn in medical microbiology, which involve in some systemic disease, we found that many types of oral bacteria can cause disease of cardio เหมือนเป็นเกาะตามพวกลิ้นหัวใจ endocarditis นะคะ so it's like the transmission from one area to the other area by like maybe go by blood or even by nerve because some bacteria can invade the nerve of the human too. So here is the summarize of the biofilm formations. After the bacteria go into the nucleus, they attach or adhere onto the surface that they want to attach. They start to produce the grooves and they grow on they grow as a community. And after that, the biofilm will like mature. We call the maturation stage. And after they like mature for a moment, there's like in that place if the bacteria send the signal to each other that this biofilm is quite very too crowded, so they will release out. So they will go to the step of the attachment to the release some bacteria out to find a new area to colonize. So here is the step of the biofilm formation. So the main thing is like once when you stay together, so you stronger to resist the environment or even to the drugs. That is the purpose of the biofilm. And also not just only live together, be stronger. Sometimes the biofilm they compose of many types of the bacteria, so they give benefit to each other. Such as sometimes you can eat bacteria A can eat some type of the chemicals and produce the metabolite, which is the food for the bacteria B. So it's like stay as sometimes it's mutualism or commensalism between each the bacteria in the community of the biofilm. So here is the example of the plaque that occur on our teeth, the creamy one here. So in this step of plaque, you can use just only your toothbrush or floss to remove out. But if you leave until the plaque accumulates other minerals such as calcium, it will be developed from just the plaque that you can remove by toothbrush or floss, develop into the calculus, which is a quite very sticky one, or หินปูนแล้วเนาะจากแค่พลาสเฉยๆ In Thai we call คราบจุลินทรีย์ for the plaque นะคะ and calculus in Thai we call หินน้ำลายเนาะ So in the step of the calculus or Thai we need the dentist to use the instrument to like scaling those calculus out นะคะ Then after that you have to clean teeth to like use for a long time. Here is the picture of the biofilm formations, the animations. Okay, that's all of the lecture about the bacterial classifications. So for this lecture and the method of microbiology, in study of microbiology will be graduate 8 point. So for each one. So for my, I have a question. So for my test, would you prefer MCQ or multiple choice or written? Multiple choice ส่วนมากคือผิดคือศูนย์เขียนเนี่ย 0.25 เคยให้มาแล้วให้ลองไปตัดสินใจดู แล้วก็ 0.25 รวมๆกันหลายๆข้อเคยมีคนผ่านมาแล้ว ให้เราตัดสินใจดูว่าจะเอายังไงปกติที่ผ่านมาพี่ออกเป็นเขียนแต่ว่าพี่ก็จะถามทุกรุ่นแหละว่าจะเอาเป็นยังไงก็ลองไปคุยกันดูในรุ่นก็ได้ค่ะแล้วเดี๋ยวเจอกันในแลบเพื่อนที่อยู่ในแลบพี่ให้มาดูส่วนมากเป็น short answer ช็อตบ้างยาวบ้างแล้วแต่อารมณ์ขอแบบ true or false true or false เนี่ยตัวดีเลยนะ น้อยมากตลอดเพราะแม่งแบบไปทีไรกูไปฟ้องทุกทีก็ไม่เข้าใจแบบว่าถ้าถูกที่เราจะถูกถ้าผิดข้อที่ผิดแม่ก็ให้ถูกเอาบางทัวร์เขาก็ได้แต่ว่าที่จะบอกว่าทัวร์เขาเนี่ยพี่ตรวจมาหลายปีละของคนอื่นนะของอาจารย์คนอื่นกลายเป็นแบบเอ้าเหมือนจะทำได้โอ้แทงผิดกันทุกคนเลยก็ลองไปคุยกันดูก่อนก็ได้ค่ะจะมีทั้ง